Hello everyone, welcome to our channel Love of Biology. Today we are going to learn about the anatomy of the dicot and monocot stem. So anatomy is the study of internal structure. So let us see first dicot stem. So it is covered with epidermis. It is the outermost layer and has a single layer of parenchymatous cells. It possesses stomata and large number of multicellular hairs, what we call stem hairs as trichomes. The outer walls are greatly thickened and cutinized. The cells are compactly arranged and do not have any intercellular spaces, and this epidermis will give protection to the plant. After epidermis, the 2 to 3 layers of hypodermis is present. So in dicot stem, hypodermis is made up of collenchymatous cells which are flexible. These cells are specially thickened at the corners against the spaces due to the deposition of cellulose and the pectin. The cells are living in the nature and may contain few chloroplasts. So it is photosynthetic in nature. And these, because of the collenchymatous cells, these are very flexible. So after this hypodermis, there is a general cortex cells. So general cortex, first let us see the diagram here. So stem hair, that is trichomes, are present on the epidermis. So cuticle also you can see here, and also epidermis. So after epidermis there is a hypodermal layer, that is collenchymatous one. After collenchyma you can see the general cortex. So after that endodermis, pericycle. So cortex, it lies below the hypodermis. It consists of few layers of thin walled, large, rounded or oval living parenchyma cells having intercellular spaces. They serve for food storage. So they will store the food and made up of parenchyma living cells with the spaces. After that endodermis, it is a single innermost layer of the cortex which separates the cortex from vascular bundles. Cells are somewhat barrel shaped and compactly arranged, having no intercellular spaces and parenchymatous. Usually they will store the starch grain, so that's why we are calling them as starch sheath. So here you can see the endodermis, and after that pericycle. So after general cortex, the innermost layer is endodermis. After that pericycle, it is made up of the layer of cells around, after endodermis, around the vascular bundles. So inside you can see the vascular bundles arranged on pericycle. It lies in between the endodermis and the vascular bundles. It is generally composed of sclerenchyma and parenchyma cells. Dead cells and living cells are present. So next, vascular bundles. They are arranged in a ring around the central pit and inner to the pericycle. These are conjoined, that means they are present on the same radius, collateral and open type, and wedge shaped. That is, cambium is present. If cambium is present, what is that? Secondary growth is present in this type of the plants. So they are present on the same radius, and laterally xylem is towards the center and phloem is towards the pericycle region. They are arranged, the bundles are arranged in a order. In between those xylem and the phloem, cambium is present, which helps in the secondary growth. Next, let us see the pith. It occupies the central portion of the stem. It is composed of thin walled parenchyma cells which are rounded or polygonal with or without the spaces. Food is stored in this region. Medullary rays. Next, so in between the bundles, vascular bundles, these medullary rays are present. These are the thin walled, radially elongated parenchymatous cells present in between vascular bundles. They store the food and helps in internal translocation of the water. So they will join the two vascular bundles. It will connect that. So here you can see the medullary rays and cambium between the xylem and the phloem. Towards the center, xylem is present, and towards the pericycle, phloem is present. They are arranged in a order. So at the center you can see the medulla or the pith region you can see. So after this is the structure of the dicot stem. So next, coming to the monocot stem, which is also similar like a dicot. So with a few exceptions, there are no trichomes, that is stem hairs, which will prevent the water loss. Those are absent here. And hypodermis made up of sclerenchyma cells. In dicot it is made up of collenchyma, but here dead cells they are made up of. And vascular bundles are not arranged in a order in pericycle, but these are present in the general cortex and they are scattered Next, chemium and the phloem parenchyma, they are absent. If chemium is present, what we call? So they are secondary growth is present, but here chemium is absent. So in the ground tissue, you can see the vascular bundles larger at the central and smaller at the peripheral. Cuticle is present without the trichomes. You see here? So there is no trichomes. And after that, epidermis and sclerenchyma, hypodermis and vascular bundles, you can see. So here vascular bundles are covered with a sclerenchymatous bundle sheath, they are covered. So this is about the monocot stem. Thank you for watching. Please subscribe my channel. Thank you everyone.